Lab 8, muscular tissue and the muscular system. We have two muscle labs. The first one, this one, we're going to look at a lot of the tissues, the microscopic anatomy, and we will start to introduce certain regions of muscles, that we're going to be identifying on different muscle models. And where indicated on the information I'm providing you in these lab exercises, there will be certain origins and insertions, or maybe even actions of muscles that you additionally have to know. So in the subsequent lists of muscles, you're to identify all of them, but some of them have particular origins and insertions or actions that you need to know. And then the other ones, I don't hold you responsible for. Okay, so the tissues, we're going to look again at a dense regular connective tissue, the histological slide, and its function is to, dense regular connective tissue forms ligaments and tendons. Right? Ligaments and tendons. Then we're going to look at the skeletal muscle anatomy. We have a particular model we often use. We're going to look at the connective tissues that are found surrounding or within the muscles. So we have, and it says identify on a model and describe the function. And sometimes the function is a little more of a description, but ultimately these connective tissues will translate forces to the tendons, and the tendons attach to bone so that it moves the skeletal system. Right, so the epimysium is the connective tissue layer that surrounds the entire muscle and continues at the ends as the tendons. So epimysium surrounds the entire muscle and continues at the ends as the tendons that are going to attach the muscle to bone. So you have a whole, it's going to outline and kind of define, let's say, the biceps brachii muscle. All right, then we're going to take that entire muscle and we're going to break it down into smaller units called fascicles. And the connective tissue that surrounds it is the perimysium. So the perimysium surrounds one fascicle. The fascicle is composed of, many muscle fibers or cells. And you're going to look at them and they're kind of on end. It's kind of like looking at a box of uncooked spaghetti down onto it. The endomysium is the connective tissue that surrounds one cell or one fiber. So how this works is that as one cell or fiber starts to shorten and lengthen, and multiple cells and fibers and fascicles are going to start to shorten and lengthen really all at the same time. But as they generate force by shortening like this, and we're going to be looking at that, it translates that force to the endomysium. And the endomysiums of all the different cells or fibers within a fascicle are all interconnected to one another. And they connect ultimately to the perimysium, the connective tissue that surrounds that entire fascicle. And then all the fascicles of that large muscle are interconnected to each other and ultimately attached to the epimysium. So that as all these cells shorten and lengthen, the force gets translated to the endomysiums, all the perimysiums, to the epimysiums, and ultimately to the tendons, and then the bone starts to move. Okay, the skeletal muscle slide. Function of skeletal muscle is to produce movement, especially the skeletal system, like locomotion of some kind, and generates heat. We're going to look at a neuromuscular junction slide where the motor neuron, the nervous system, meets the muscle tissue. We're to identify the following structures: the motor neuron axon, the axon terminals, and the skeletal muscle fibers. And so we'll look at that on the slide. We have a motor neuron. It kind of looks like a spider. And we will see more of these neurons in the nervous system and neurology in the next chapters. We have one more muscle chapter, but we're going to, after that, we have exam 2. And then we go into the nervous system. So we're going to look at these different types of neurons, sensory neurons, motor neurons, in more detail in the next chapter. But because it's such a significant part of muscle contraction, because every skeletal muscle, cell has its own nerve and blood supply to it, is that it's a critical part of the physiology of muscle contraction, how our nervous system stimulates muscle contraction. So we're going to look at the motor neuron axon, axon terminal, skeletal muscle fibers. Okay, then we have the muscle physiology. Identify the following structures on all the muscle fiber models and describe functions. Structures are in correct order of muscle stimulation. So, going back to this neuromuscular junction slide kind of concept, we have the nervous system telling these muscle cells to contract in some way, shape, or form. So this impulse or this message is going to travel down this motor neuron, and it travels down to the axon terminal. It's like little fingertips of a motor neuron. And in those fingertips, there are these vesicles that store what's called neurotransmitters. And the neurotransmitter is a chemical compound, a signal that when released from this neuron will travel across the slight gap between the neuron and the surface of the muscle cell and look for a receptor on the muscle cell for that chemical compound to attach to. And when it does that, then it stimulates a signal within the muscle to travel in through that muscle around the surface of it, down what's called these transverse tubules to the interior of the muscle cell, so that we start a series of physiological events that ultimately produce this shortening of the muscle fibers. And so we go from the macro anatomy, which we've been talking about up above, to now the micro and physiological, sequence of events for muscle contraction. So we start with the motor neuron axon, and axon terminal would be the next structure in order. Acetylcholine to the sarcolemma, transverse tubule, sarcoplasmic reticulum, actin, and myosin. Right. We're going to put the function in there and match these functions to it. All right, so A, the thin filament with a binding site for cross-bridge formation. And I put in parentheses, this is where troponin and tropomyosin complexes are found. That would be on actin, on the actin molecule. So A goes with actin. That's kind of handy there. B, the thick filament that binds to actin and produces a power stroke. That's myosin. Actin and myosin are these contractile proteins. One of them is kind of the skinny one. That's the actin. The myosin is rather chubby, and it looks like they have like oars on the surface or maybe golf club heads. And that to produce tension within the myosin needs to make physical contact with actin, and then it kind of rose toward the center of what's called the sarcomere, and we get muscle tension being produced. Okay, C, the neurotransmitter that binds to a receptor on the sarcolemma. That is acetylcholine, or ACH. D, the impulse, carries impulses to the deepest region of the cell. Those are the transverse tubules. E, contains acetylcholine receptor sites. That's the sarcolemma. F, stores and regulates calcium ion release. That's the sarcoplasmic reticulum. G, carries stimulation away from the spinal cord. That's the motor neuron and its axon. H, has vesicles that release acetylcholine when stimulated. That is the axon terminal. So, we used to have some like laptops in the lab, and we had some programs that were loaded up to it that I guess we're not using anymore, now, especially post-pandemic. But we had some videos that you could look at in the lab that would put these kind of in sequence so you can see them. In your lecture classes, I know in mine, I have about 3 or 4 videos that break this down into, subsets, and they're from your textbook. There's a lot more detail in parts of the sequence here, the physiological sequence, starting with the motor neuron all the way to generating a force, between the actin and the myosin at the end, but this is kind of the short version. And if you have this down, then, being able to probably deal with about any lecture class you have, the sequence of events, the physiological events will be much easier. Okay, so now we're going to move on to the skeletal muscles that we're going to identify, and then you can see which origins, insertions, and actions you need to know. So these are the head and neck muscles. Over to the left-hand side, vertical column, we have the muscles. So you're to identify all these muscles: orbicularis oculi, orbicularis oris, zygomaticus, mylohyoid, digastric, temporalis, masseter, sternocleidomastoid, and splenius. And I thought I had a notation for what the little, I think the asterisk just means that you need to know something else, like an origin insertion, or maybe even an action for those muscles. Okay, so we have orbicularis oculi. That's the round muscle of the eye. And its action is to close the eye, blink, or wink. Orbicularis oris, the circular muscle of the mouth, closes your lips and mouth, protrudes the lips. So you can call it the kissing muscle, or as you're chewing, you generally purse your lips so food doesn't fall out of your mouth. So you can consider it that way. Zygomaticus. There is a minor and a major, but we just generally point to the major and just call it zygomaticus. Right now, this has your first origin insertion that you have to know. It originates on the zygomatic bone, thus that's why it's called zygomaticus, and it inserts on the corner of the mouth. Essentially, it inserts on at the corner of orbicularis oris, and it raises the angle of the mouth, as in smiling. Under the mandible, on the undersurface, under your jaw, by your neck in that area, there are two muscles you have to identify: the mylohyoid and the digastric. The mylohyoid is a deep muscle with fibers coursing in the direction opposite to what's called the digastric. The digastric, or the anterior digastric, is kind of very small and superficial to the mylohyoid. So I'll show you a picture of what it looks like. You just need to identify those two. Okay, then we have temporalis. Temporalis is named for the bone that it originates on. So it originates on the temporal bone and inserts on the coronoid process of the mandible. And it elevates the mandible, as in closing the mouth. The masseter is a much larger muscle. It's closer to your ear, from your temporomandibular joint, down to the undersurface of the mandible. Now, I call it the pit bull muscle because it's that big muscle right, deep to your cheek tissue there. And you just have to know that it elevates the mandible as well. You can clearly see the origin and surface and insertion of temporalis, but not so much on masseter. You can see it for sure, though. All right, then we have sternocleidomastoid. That muscle is named for the two origins and one insertion. It originates on the sternum and the clavicle and inserts on the mastoid process. And then we have the splenius muscle. That's found on the back of the head or neck area. It's going to be a very vertical muscle. It's one of the erector spinae muscles. Again, you just have to identify it. So these muscles to the anterior aspect, and then to the only one to the posterior aspect. So, and I'm just looking at this, don't worry about the asterisks there. Yeah, I know previously I did not have these already filled in for you, and so just disregard those asterisks. They really mean nothing in particular. But you do have to identify all the muscles, just to be clear. And of those identified here, of the origin, insertion, or action, you need to know those for that particular muscle. Okay, then we're going to go to some of the torso muscles, and then we'll do stuff on arms and legs and the pelvis next week. Okay, so we have serratus anterior. You just had to identify it. Serratus anterior is found sort of just inferior to your armpit along your ribs, and it has a serrated knife edge kind of an appearance, like a zigzag. Pectoralis minor originates on some of the ribs here on the anterior aspect of your chest, and travels to the coracoid process of the scapula. So it's going to be deep to pectoralis major. Okay, on the posterior aspect, the big superficial muscle of the upper back is trapezius. Just identify. If we pull trapezius off and look at the rhomboids, they are in a rhomboid shape. You have to know what a rhomboid is, but it's kind of like having a domino. So you know how it's taller than it is wide. And if you kind of sat on it and made it kind of tilt to one side, so to speak, that's a rhomboid. You do have a rhomboid minor and major, but we grouped it together as a rhomboid. Then we have levator scapulae. Levator scapulae, again, is deep to trapezius. So we have to be looking at what's called the deep side of the muscle model, not the superficial side. And I'll explain that when we get to be looking at the models, or maybe just at the end of this, I'll kind of explain it. So levator scapulae elevates the scapula, right? It originates in some of the transverse process of cervical vertebra and then attached to the scapula itself, right? So it's going to go from vertebra in the neck and attached to what's called the superior angle, the most superior medial aspect of the scapula. So when it shortens, it elevates your shoulders. Right? All right, rectus abdominis, one of the abdominal muscles, straight up and down and in the middle. Its origin is the pubic symphysis and inserts on the xiphoid process. It flexes the torso and compresses the abdomen. Now, technically, we have 4 abdominal muscles. We have rectus abdominis and transverse abdominis. That rectus abdominis is superficial, transverse abdominis is very deep, and they run perpendicular to one another. But we usually don't have you identified transverse abdominis because you'd have to take all these models apart, and they're generally a nightmare to put back together. Right? To the sides, to the right and left, we have our oblique muscles, and they're layered on one another. They too their fibers run perpendicular to one another at a 90-degree angle, but the muscles tilt over on, obliquely, kind of at a 45-degree angle, so that they're not running straight up and down. They're running kind of from your shoulder down to your umbilicus or your xiphoid process kind of out to your hip bone. So they're going to be traveling in an oblique angle. The most superficial one is the external oblique. The internal one, or the one deep to that, is the internal oblique. And then on the back of the spine, paralleling the spine, and really splenius is part of this group, but we'll look at this group of muscles that are very close to the spinous processes and traveling superior and inferior. Those as a group is called the erector spinae muscles. And they help extend your torso. So they would do a back bend on you. Or if you bent forward at the waist, the erector spinae muscle would contract and bring you back to anatomical position. So what I started to say before is that, when we talk about these that are superficial and deep, the reason why I say that, and then I'm going to switch to the images, is that the models, with the exception of the arm and leg models, I call them arms and legs on sticks, is that when we're looking at the torso models, in general, when we're looking at even like the great big muscle men models, because they're huge, they're like, 5-6 feet high, is that one side of the model shows you all the muscles that you can see when you take the skin off. And these are what's called the superficial muscles, the first layer of muscles that you would see. But virtually everywhere in the body, we have layers of things. It's not just one layer, it's a middle layer and a deeper layer, and sometimes a third and fourth layer of muscles. So one side is going to show you all the superficial structures. The other side shows you what the muscles you can see when you take that top layer off. All right. So it doesn't mean these muscles aren't, that not the, if they're the same side to side, but you have one model that can show you what is deep to the most superficial muscles, and it, makes one model do kind of double duty. Okay, so let me switch over to the images and let's get started. Oh, by the way, before I do that, there is, oops, I clicked on the wrong one. Hold on. Down below in because we are in lab 8. I was just here in the document, of all of the different lab exercises. Below that, this video is good. It looks at all the muscle anatomy and cellular anatomy, the models. I did these videos, or I made these videos on, a phone. Some of them were on my phone, some was with someone that was helping me just before we locked down for the pandemic. I had like 2 or 3 days to come in and try to make videos and take whatever I was going to need for 6 months to, it turned out, about a year and a half. And these videos are actually pretty good because I'm in the lab walking around the muscle models and looking at things, and I'm talking while I'm making this video, so hopefully, it helps you. And that way, even if you're at home doing this, you can see a little bit better about what these models actually look like so that the images make more sense. Okay, so over to the images. Let's start with, here we have a histological slide. All right. This is dense, let me back up. This is skeletal muscle. The dense regular connective tissue is generally in a kind of an orangey kind of a color, but it does have very parallel fibers. All right, so this is skeletal muscle. It generates heat, it moves the skeleton. We will technically, if we look at these fibers, we can see them very, very parallel to one another. And if we magnify this a little bit more, you can see all the the multinucleated nature of these muscle cells, and you can see these lines. Now, some of these you can actually see. So here we have these muscle cells and fibers, of course, and they're parallel to each other. They have a little connective tissue in between, kind of running on, again, an oblique angle here. And you can just about see, so running perpendicular, which means 90-degree angles to the length of the fiber, are these smaller little, it looks like you took a pencil and drew these lines. That represents the region from sarcomere to sarcomere. And the sarcomere is the repeating functional unit of skeletal muscle. Those are what's called striations. These take up some stain, and you can see these little pencil lines traveling from one side of this muscle cell or fiber to the other. Those are striations, and that represents the end of one sarcomere to another sarcomere. And those sarcomeres repeat all the way down the length of this entire muscle cell or fiber, and then you have many, many, many thousands of them. This, which isn't the best picture in the world, but this is, if we could get it to take up a little more stain, is kind of orangey in its tint. And this is dense regular connective tissue. Very parallel collagen fibers. They form functionally tendons and ligaments. Tendons attach muscle to bone, ligaments attach bone to bone. Okay, then we have the neuromuscular junction here. The background tissue here in purple, but sometimes they're in more of a red color. That represents the skeletal muscle, because all skeletal muscle cells have to have their own nerve supply to them. Now, this black here that looks kind of like spider legs, those are the motor neuron axons. They terminate in these little round regions here called the axon terminals. So it's kind of like their fingertips. And if we magnify this, and actually you can somewhat see them here in this little cluster here, we can see some little vesicles in there. They're very dark staining. They sit on top of each other, but you can somewhat see these little balls there. Those would be the synaptic vesicles. And vesicles, are these phospholipid bilayer balloons, so to speak, that store something inside. And these vesicles here store acetylcholine or some kind of neurotransmitter. But in this case, it's acetylcholine. When an impulse to contract, this electrical kind of impulse travels down these axons and arrives at these fingertip areas called the axon terminals, those vesicles will merge with the membrane and release the contents exocytotically of that acetylcholine. And then it binds to receptors on the skeletal muscle tissue here in the purple background. And now the skeletal muscle creates an electric signal or an impulse, an action potential that travels around the muscle, down these transverse tubules, to the sarcoplasmic reticulum. Sarcoplasmic reticulum releases the calcium. Calcium binds to troponin and tropomyosin. Tropomyosin uncovers the binding site on actin, and now myosin can form a cross bridge, and then you get muscle shortening. So here we're at a neuromuscular junction. These are the axon terminals. Here's the motor neuron axon. It's actually the black lines are actually, the motor neuron axons. And at the ends, the tippy dippies there, the fingertips of this, that's where the axon terminals are. And they contain neurotransmitters. Or they contain the vesicles that store the neurotransmitter. Okay, here is one of the models. This represents, one skeletal muscle fiber or cell. You can use those terms interchangeably. We can see a couple different features on here. All right. First of all, 1, 2, and 3 represents the motor neuron. So here, 1 is pointing to this yellow region in the center. And then you have these, it looks like there's kind of like a Pacinian corpuscle. You have layers surrounding this interior yellow region, right? That's part of this motor neuron. And the impulse to contract is going to travel down this whole big structure, number 1. And it arrives at the axon terminals represented here in 2. Now it's cut open. You can see some mitochondria in there. You can see these little white little balls there. Those are the synaptic vesicles storing its acetylcholine. All right, so when the impulse travels down on number 1, it arrives at 2, and those little white balls merge with the membrane, all right, and releases the acetylcholine into this gap area. And then it's going to bind to the surface of this skeletal muscle cell or fiber, so that now an impulse travels within that cell. You have over to the left-hand side, you can just barely see a white layer here surrounding, if we turn this model around, you'd see it more distinctively, but you can just barely see because they cut open this model so you can see the interior. That would be the endomysium. Now, if I look to the bottom here, you have kind of a pink membranous region here. That is the sarcolemma. So the sarcolemma is just a fancy word for the plasma membrane of a muscle cell or fiber. And it's in this kind of medium pink color here. It surrounds the whole thing, but that's where we see it down here. While we're here, I'm just going to point to it all. All right, that pinches inward, right, the plasma membrane, that sarcolemma, and forms these tubes, so that the impulse generated on the surface of the pink here will now travel within the interior of these blue tubes, because you can see that pink color continues to the interior of these blue tube system, and those blue tubes are the transverse tubules. So they're going to carry an impulse from the surface of this muscle cell or fiber in the side to the interior of these tubes to the interior regions of this whole cell. They sit on top of the light peach network of tube systems called the sarcoplasmic reticulum. And then the sarcoplasmic reticulum that stores the calcium will release it. That calcium will bind to the thin and, what's called the troponin tropomyosin molecules that block binding sites on actin. Actin is either the skinny fibers here, the little skinny red fibers, and the myosin would be the thicker red fibers. There should be another model that gives you a slightly better idea of this that I like to use. So hold on. All right, so 1, 2, 3, we're looking at like the neuromuscular junction on a model. We had the motor neurons, synaptic vesicles, storing their acetylcholine, the sarcolemma. Right, this is that same model, but it's pointing to the blue tubules versus the light peach network there. Sarcoplasmic reticulum that stores and releases calcium, and the transverse tubules, carry an impulse to the interior of the cell. Here we have, darker and thinner, or thicker and thinner red lines. Those are the myosin and the actin filaments. The myosin are the chubby fibers, the actin are the thinner fibers. So the darker red is the myosin, and the lighter red, skinny ones are the actin. This is looking at a model that represents the same thing. It's just a different manufacturer. But this lets you see the outer connective tissue layer, number one. That would be the endomycin that surrounds one cell or fiber, right? And two is pointing to the sarcolemma, the plasma membrane of that muscle cell. All right. 1, 2, and 3 is again pointing to the similar nerve structures or components where we have a motor neuron axon, the axon terminals. 3, represents the binding sites for some of these neurotransmitters or acetylcholine in this case. So that's the receptor site on a sarcolemma. This is another type of model. This, and there should be hopefully another one, which I like even better. But what's good about this is showing you in light blue network here instead of the light peachy. This is a bigger model. That is the sarcoplasmic reticulum. And the green tube systems on top of it, these would be transverse tubules. On the other model, those transverse tubules were in a darker blue, and the sarcoplasmic reticulum was in like a lighter peach. This is just, a different type of a model representing the same thing. But it also lets you see the difference between the skinny fibers and the darker fibers of the myosin and actin, at 1 and 2. So 1 represents the myosin, those are the thick filaments, and actin are the thin filaments, number 2. This is I like this one because it's just brighter, I guess, but here we have, instead of in light peach or in whatever it just was, light blue, the sarcoplasmic reticulum is in this bright yellow network, right, which stores and releases calcium. The transverse tubules on this model are in the green here. So they carry the impulse of transverse tubules from the sarcolemma, because the interior of these transverse tubules actually is sarcolemma, as this impulse can travel down these tubules. The other thing I like about it down on below are the thicker and thin gray lines here. And I think these are a little bit easier to see because they're spread out a little bit. But right here over to the left-hand side, you can see the thinner dark gray, not the light gray. The light gray is all the, like the cytoplasm, cytoplasm, within the cell. But that's the light gray. But the darker little lines, are the actin filaments or the myosin filaments. Thin dark gray lines are actin. Thicker, like toward the center, dark gray lines are myosin. Okay, so the light gray is technically, did I say cytosol? Cytoplasm is everything between the nuclear membrane and plasma membrane. I have a bad habit of using those interchangeably, so my apologies. Thin gray, dark gray lines are actin. Chubby dark gray lines would be myosin. I like this model for that also. Okay, as we mentioned before. And now we're going to start looking at muscles. So here, if we could rotate this vertically, number 1 is looking at the circular muscle of the eye, 2, the circular muscle of the mouth, and 3 is looking at that sternocleidomastoid, or what we call the SCM. All right, so we have orbicularis oculi, orbicularis oris, and sternocleidomastoid. Right, so, as I said before, when we were going, filling in these lab exercises, is that when indicated, you need to know actions, maybe an origin and or insertion. And orbicularis oculi is an easy one. So is orbicularis oris because they're circular. So 1 closes the eye and winks. 2 purses the lips, and your kissing muscle. Sternocleidomastoid, you need to know because it's easy to see these. The 2 origins, the sternum and the clavicle, and you can see those 2 bones there. Look at where the muscles are starting from and where they're going. So it's beginning here down in the chest, and it's traveling to the mastoid process on the temporal bone. You don't have to know the action. You just have to know the 2 origins and the 1 insertion. So, I'm sorry, I'm just going back again. So I could ask you, and then this is where I can give you examples of questions. Number 3, identify that muscle. Sternocleidomastoid. Identify the origin, and you'd be multiple choice, sternum and clavicle, or clavicle and sternum. Or I could ask you, identify the insertion of 3. That would be the mastoid process. Remember, the origin is usually the end of the muscle or the bones that the bones that this muscle attaches to. The bones that don't move or have very little movement is considered the origin. The other end of the muscle, the bone that it attaches to, is generally going to be the bone that moves the most. And so in this case, your sternum and clavicle are going to stay stationary, and your head is going to move because it's inserting on the mastoid process of the temporal bone. Excuse me. Okay, now we're going to have a sideways view. Here we have number 1, temporalis. 2 is pointing to this very large muscle here. That's your masseter, the pit bull muscle I call. And 3 is pointing to zygomaticus. So here the temporalis, muscles that end in ALIS. ALIS in Latin means pertaining to. So temporalis means pertaining to the temporal bone. It originates on the temporal bone and inserts on the coronoid process of the mandible so that it elevates the mandible in chewing. 2 is the masseter. It does that also. This yellow gland here is your parotid gland. So if we could take that off, you could see how large that masseter muscle is. Elevates the mandible. And zygomaticus originates on the zygomatic bone. Again, if we move number 3, we would see that a little bit better. But inserts here on the corner of the mouth. And it elevates the corners of the mouth as I'm smiling. The zygomaticus is going to be a muscle of facial expression. Temporalis and masseter, because they're involved in chewing, will be muscles of mastication or chewing. Okay, now we're looking at the undersurface of the jaw. I said that there were 2 muscles you need to be able to identify. The digastric or anterior digastric, which is superficial and rather small, that's number 1. And the mylohyoid here is pointing to it number 2. But even over to the right and left, you can see these fibers going out toward the ears. That is really a better example of mylohyoid, but I guess I'm going to have to use that number 2. You just have to identify them. But what they do is that when they contract, they open the jaw. I mean, the jaw will depress, or the mandible will depress, so that you can put food in your mouth. Okay, now we're looking at the torso model. I'm going to move kind of in different directions. But this arrow is pointing to the zigzaggy muscle that's here kind of in the armpit area. That would be serratus anterior. Now we're looking at the back of the skeleton, or the muscle man, I should say, not the skeleton. And we're looking, when you can see bone here, here's the head over to the right-hand side, and the left arm is extending up here at 12 o'clock. All right, so you have this muscle that's very parallel here to the spine. That one is called splenius. 2 is the muscle that's going from the cervical vertebra to the scapula. That's levator scapulae, elevates the scapula. And then 3 is pointing to the larger of these 2 rhomboid muscles. So you just have to identify that as muscle as a rhomboid. Splenius, levator scapulae, and rhomboids. So remember, as indicated, you need to know origins, insertions, and actions of some of these muscles. I'm not going to go back through all of them as we're going through here because you'll fall asleep. Okay, so remember I said before that, when it comes to these models, we have a superficial side and a deep side. So the muscles we just looked at in the previous view, it's the same model. We're looking at this great big muscle man and we're looking at his back. All right. This side is the deep side, right? Where we've taken off what's called trapezius, which is down here on the other side. You can see the little spinous processes here of the vertebral column. All right, below that, that's your trapezius. So that's going to be the superficial side. These are the muscles we can see when we take this big trapezius muscle off. So that's your deep side. Right. The other side, which is going to be this model's right-hand side, is going to be the superficial side. So again, if we could rotate this is huge. This is trapezius from the neck area out to the shoulder here, all the way down. It looks like it's three separate muscles. That is one muscle. That is trapezius. Below that, this big superficial muscle, that goes all the way down essentially to your pelvis, to the iliac crest. That is latissimus dorsi. Trapezius. So superficial muscle of the upper back is trapezius. Superficial muscle of the lower back and massive is latissimus dorsi. Some of those, the trapezius we're looking at now, latissimus, we'll look at the next lab, but at least we saw a picture of it. Okay, here we're looking at the anterior or the front of this great big muscle model. Again, we have a superficial side, which would be here where the number 1 is, and that chest area there, that would be pectoralis major. On the other side, and they actually show you where they cut the muscle, right? Now we can see structures or muscles below, or that this pectoralis major muscle is obscuring your ability to see. So below that number 2, originating from some of the ribs here in the chest area, thoracic ribs, is going to be pectoralis minor. And that originates on ribs and inserts on the coracoid process here on the scap. 3, because I can't read. 3 is pointing to rectus abdominis, or that's the six-pack muscles on some people. The vertical muscles midline that originate on the pubic symphysis and insert on the xiphoid process, can flexes the spine, compresses the abdomen. Here we are looking at that great big model, but now it's upright, and again we can see to the model's right, but our left we can see the superficial muscles, and on our right side we can see the muscles that are deep to it. Right. So rectus abdominis is the one here right in the center. This would have been a very good view. Over to the right here at number 2, since we're on the deep side, that would be the internal oblique muscle. And on our left over here at number 1, the superficial side of the muscle model, that would be the external oblique. This is the model that represents the 3 different connective tissue layers. One is to the most superficial and the outermost layer, so that would be the epimysium. Remember, the epimysium surrounds the entire muscle, continues at the ends as the tendons. Tendons attach muscle to bone, ligaments attach bone to bone. 2 represents the connective tissue surrounding one fascicle. And you can see how now, like this muscle is being divided into little subsections here, here, and here. That represents one fascicle. So that would be the perimysium. The perimysium surrounds one fascicle. And then 3 is pointing to the deepest layer. That would be the endomysium, the connective tissue surrounding one cell or fiber. Okay, so epimysium, perimysium, endomysium. What you can't quite see, and it would have been nice if they had a slightly better color change, is this epimysium is kind of a pretty darkish white color. The next, it's like a little step down, is the connective tissue surrounding, each of these kind of launch, horizontal clusters of muscle cells. Those are fascicles. And there's a slight step down, but this tissue here at 2 is the perimysium. Then there's another little step down here. So you see the connective tissue surrounding one particular cell. So it's a little difficult to see this way, but I'll use this picture just without the index card. Epimysium, perimysium, endomysium, and be able to recognize a reasonable definition of those tissues. Okay, and so that is the end of Lab 8.